This is the first lecture in body fluid. We will take in this lecture the renal function and renal physiology. This lecture is by Dr. Fahim. Each kidney contained approximately 1 million to 1.5 million functional units called nephrons. We have two types of nephrons, the cortical nephrons and juxtamedullary nephrons. Cortical nephrons, it's about 85% of all nephrons. Situation in cortex kidney. The primary function is responsible for removal of the waste product and reabsorption of nutrients. The second type of nephrons is juxtamedullary nephrons, longer Henle loops. The primary function is concentrating the urine. The ability of the kidney to clear the waste product from the blood and maintain the body's essential water and electrolyte balances is controlled in the nephron by renal function, depend on four types of process. First one, renal blood flow. Second, glomerular filtration. Third, tubular reabsorption. And the fourth, tubular secretion. Renal blood flow. A renal artery supplies blood to the kidney, where receive 25% of the blood pumped by the heart. Blood enters to the capillary of the nephron through the afferent arteriole, then through the glomerulus, then to the afferent arteriole. The varying size of arterioles create difference in hydrostatic pressure. This difference in hydrostatic pressure play a role in maintaining glomerular filtration and maintaining glomerular capillary pressure, and finally renal blood flow within the glomerulus. The smaller size of the afferent arteriole increases the glomerular capillary pressure. Before returning to the renal vein, blood from the afferent arteriole enters to the peritubular capillary and vasa recta through the cortex and medulla renal tubules. The peritubular capillaries surrounded the proximal and distal convoluted tubules for reabsorption of the essential substance from the fluid and proximal convoluted tubule and forming urinary composition in the distal convoluted tubule. The vasa recta is located near to ascending and descending Henle loop in juxtamedullary nephrons, where major exchange of water and salt takes place between the blood and the interstitial medulla. This exchange maintains the osmotic gradient salt concentration in the medulla, which is necessary for renal concentration. The total renal blood flow is approximately 1,200 ml per minute, and the total renal plasma flow ranges between 600 to 700 ml per minute. But the values of renal blood flow and the renal function test depend on body size. The second process gives the kidney the ability to do its function, its glomerular filtration. The glomerulus consists of coil of 8 capillary loops called collectivity capillary tuft, located within the Bowman's capsule and forms the beginning of the renal tubule. The glomerular filtration is non-selective of plasma substance. With molecular weight less than 70,000 kilodalton, but there are several factors influence the actual filtration process. The structure of the capillary walls and Bowman's capsule, hydrostatic and oncotic pressure, feedback mechanism of renin-angiotensin-aldosterone system is called RAS. The cellular structure of glomerulus. Plasma filtrate must pass through the three cellular layers. First, the capillary wall membrane is called endothelial membrane or endothelial cell. The endothelial cell of this capillary wall differs from the other capillary wall because it contains pores. These pores increase the capillary permeability, but don't allow to passage the large molecule and blood cells. The second layer is basement membrane basal lamina. Its major extraction of large molecule occurs through the basement membrane. Visceral epithelium of Puma capsule are called podocyte. The same membranes covering the filtration site formed by podocyte of the inner layer of Puma capsule. Glomerular pressure. Hydrostatic pressure forming from the smaller size of the afferent arteriole, and the glomerular capillaries increase the filtration process. Hydrostatic pressure is important to overcome the opposition of pressure from the fluid within the Puma capsule and the oncotic pressure of unfiltrated plasma protein in the glomerular capillaries. Increasing or decreasing the size of the afferent arteriole make autoregularity mechanism within the juxtamedullary apparatus. Dilation of the afferent arteriole and constriction of afferent arteriole when low blood pressure prevent decreasing of renal blood flow and prevent increasing the level blood toxic waste product. But an increasing blood pressure may constriction of the afferent arteriole to prevent overfiltration or damage of the glomerulus. Renin-angiotensin-aldosterone system controls the flow of blood within the glomerulus. The system responds to change in blood pressure and plasma sodium content, which monitored by juxtaglomerular apparatus, which consists of juxtaglomerular cells in the afferent arteriole and macula densa in the distal convoluted tubule. Low plasma sodium content decrease the water retention within the circulatory system, resulting in decrease the blood volume, so decrease the blood pressure. When macula densa sense such this change, the other cascade of the reactions within the RAS occurs. Renin is enzyme produced by juxtaglomerular cells, which react angiotensin to reduce innate hormone angiotensin 1. Then angiotensin 1 passes through the lungs and the angiotensin converting enzyme ACE is an enzyme make conformation and convert angiotensin 1 to active form angiotensin 2. Angiotensin 2 corrects the renal blood flow. Angiotensin 2 causing vasodilation of afferent arteriole and vasoconstriction of afferent arterioles, stimulating reabsorption of sodium in the proximal convoluted tubules, releasing two types of hormones. Firstly, sodium retaining hormone called aldosterone by adrenal cortex. It's working on sodium reabsorption from the distal convoluted tubules. And the other is ADH, antidiuretic hormone, by hypothalamus. It's working on water reabsorption from the collecting duct. The secretion of renin decreases when sodium content increase, systemic blood pressure increase too. So angiotensin 2 produce constant pressure within the nephrons. Note, every minute, 2 or 3 million glomeruli after 120 ml of water containing low molecular weight substance filtrated. And this filtration process is non-selective. The only difference between the composition of filtrate and the plasma, or filtrate and the blood plasma, is absence of the plasma protein in the filtrate because this protein is bounded to cell, bounded to substance, so the size is big and prevented to filtrate it. Analysis of the fluid that leaves the glomerulus has specific gravity 110. This ultrafiltration of the plasma turns to the finally urinary product. When we see this specific gravity and the urine, that must know the kidney lose the ability for reabsorption the essential material for the body, and the kidney lose one of its functions. We have diagrammatic in this slide. Show us what the rats do. When the blood pressure or low sodium content is low, so some macula dense and juxtaglomerular cells sense that, so releasing the renin secretion. When the renin releasing react with angiotensin, which convert angiotensin to angiotensin 1. Angiotensin 1 go to lung and be activated by angiotensin converting enzyme. To the active form, it's angiotensin 2. Angiotensin 2 make, as we say before, make vasoconstriction for efferent and vasodilation for afferent, and make sodium reabsorption from proximal convoluted tubules. Then stimulate releasing the aldosterone and ADH. Aldosterone, as we say, work on sodium reabsorption from the distal convoluted tubules, and ADH work on water reabsorption from the collecting duct. But angiotensin 2 only work on sodium reabsorption from the proximal convoluted tubules. And now we finish our lecture with my best wish for success, Dr. Rida Fahim. Thank you.